All right, everybody. So today we have Dr. Michael Roberts with us. He has a PhD in exercise physiology, and he is a professor at Auburn University. So welcome, Dr. Roberts.
Thanks, Dave. Thanks for having me.
Sure. So we got in contact through Cody Hahn, who was recently on the podcast. And with that one, with all of these, I start off just kind of mentioning the charity that we're donating to. And so for today, we're going to be sticking with Operation Smile. And for people who don't know you, I know we talked a little bit, you haven't been on a lot of podcasts yet, but, Dr. Hahn was singing your praises. So can you give us a little bit of your feedback and how you got into this?
Sure. So I started, I guess my formal education was at Baylor University. So that's in Waco, Texas. I did my undergraduate degree in biology and minored in chemistry. So you're in dentistry. I was actually pre-dental studies and shadow dentists, and just that didn't grab me so much. But towards the end of my undergraduate tenure at Baylor, Dr. Rick Kreider, who's at Texas A&M University now, he established his lab at Baylor University, and he was examining the effects of different diets and exercise on weight loss and postmenopausal women, and he started getting into the biology of that. So he was taking blood. Soon he recruited another professor, his name is Dr. Darren Willoughby, who started taking muscle biopsies. And I liked exercising while I was an undergraduate. And once I saw the connection between biology and really human physiology with exercise and nutrition, that's when it all kind of clicked for me. So I stayed on at Baylor under Rick as a master's student. I was there until 2006. Then I went and got my PhD at the University of Oklahoma, where I delved a little bit more into the molecular techniques in terms of taking blood and human muscle biopsies. And then after that, I did a postdoc fellowship at the University of Missouri with Dr. Frank Booth, who is really one of the pioneers in the exercise molecular biology world. And I ended my postdoc in 2013, and then that's when I came to Auburn. So I've been here at Auburn University since 2013.
Nice.
Yeah.
So for somebody who is so, really like delving into the science of all this, because I would imagine it could be frustrating when you see that 95% of the purveyors of fitness information really don't have much of a scientific background. A lot of, misconceptions are put out there and just over and over again. Does that frustrate you, or do you kind of just like put your head in the sand and try to ignore it?
No, honestly, I embrace it. I see it as job security, as
Right.
a myth dispeller. I mean, I think I was telling a student the other day, I think seeing the way that fitness or the fitness world has taken off in social media with podcasts, with just mainstream publications. I mean, I'm getting requests from Auburn University as an expert trying to disseminate, a research article which discusses, is it good to fast in the morning when you exercise, right? And they post that on the first page of the website. But I say all this because I think really this is kind of, in my opinion, becoming one of those eras where exercise is in vogue, right? I mean, you see a lot more interest. I think that social media has really sparked a lot of that interest. And even though, some of it's kind of what it is, right? It's not great information, or you may call it bro science. I mean, to a certain degree, I actually respect and embrace that, because a lot of what we do here in the lab, right? People that are in the trenches, whether they're working with athletes or they're competitive bodybuilders or physique competitors, right? They don't see the validity in a lot of our study designs. And I kind of like to hear what those people say, right? In terms of dealing day to day with clients or with athletes and things like that. So, I mean, to answer your question, I think it's a beautiful ecosystem. And while there is some minutia, right, that you have to deal with, I mean, I think just the fact that exercise is becoming so popular is a net win.
Yeah, I mean, that's a good point. I mean, you could look at a lot of people hate on CrossFit, a lot of people hate on Planet Fitness, a lot of these things, but at the same time, at least it is getting people exercising. And, something I often talk about in this podcast and in other videos I do is unfortunate. I mean, I don't know if it's unfortunate or fortunate, but you can do a lot wrong and still make progress, which is why you have people who look fantastic who really aren't that knowledgeable because they're just consistent over time. So while it's better to put out good information, I think it is also good to have anything that's getting people more active in general.
Sure, absolutely. Yeah.
So one of the things Cody and I had started talking about was genetic variance in response to hypertrophy and just response to exercise in general. So I know you know a lot about that. In terms of weight training specifically, could you talk to us a little bit about some of the biggest genetic factors that we see that will affect somebody's ability to grow muscle?
Yeah, so one of my lab's interests, and this is what Cody Hahn was doing when he was a PhD student here, was trying to determine molecular factors, if you will, that differentiate high anabolic responders versus individuals that really don't grow muscle in response to weeks or months of resistance training. And I have to give credit to Dr. Marcus Baumann, who's actually at UAB, so he's in Birmingham down the road. He was really the first researcher that started addressing this question. So a lot of our studies were really inspired by his original studies in the mid-2000s. But the idea is that we take a group of people, and we've done 2 of these studies now, so we've taken 2 groups of people, and we train them for 6 to 12 weeks in the weight room. Everybody gets the same relative load according to their strength. And we do pre and post intervention assessments, and we look at changes in things like muscle thickness using an ultrasound. Now, some people, they'll see this robust increase in muscle thickness, or we also take biopsies out of the leg, and they'll show this increase in muscle cell size. So we call those the high responders. What's fascinating to us is that some people show no meaningful response, right? So those are your low responders. From there, since we have the tissue, what we try to do is we try to look at biomarkers that exist at baseline that could be explaining, right, why one group responded whereas the other didn't. And then we also look at the pre to post change in those biomarkers in muscle. And so we haven't done formal genetics testing. Like, you can probably go onto PubMed and look up genome-wide association studies for any particular trait like BMI. That's where the FTO SNP, the obesity risk allele, pops up if you look at BMI and genome-wide association studies. But the idea there is that researchers will get a lot of people, a lot more than just 100 people. They'll get thousands of people, and they will test all of these polymorphisms from a blood sample or a saliva sample from buccal cells, and they'll say, these genetic variants are linked to the phenotype. So we haven't done that, but what we have done is we've looked in muscle, and we found a couple of biomarkers which may be suggestive of why some people respond to training versus why others don't. And the main marker in muscle seems to be ribosome content. Mm-hmm. So for listeners that aren't aware of the physiology there, ribosomes are the macromolecules which facilitate muscle protein synthesis. So to us, that kind of makes sense. If you see an increase in what's called ribosome biogenesis, or the creation of new ribosomes with resistance training, then according to our data, as well as others like Marcus Baumann, you probably will be a high responder. Whereas if you don't see an increase with training, then you may not be a high responder.
Yeah, it's interesting because, anybody who's taking like cell biology, and, we all have that like generic idea of the cell. And it was never really taught that there would be variations in the number of organelles that are in there. I mean, it makes sense, like there's variations in pretty much everything. Sure. But to think that one factor can have a significant impact is pretty interesting. Yeah. I'm curious, what is the longest study you've seen where somebody was not responding at all? I mean, because a lot of these are shorter term.
Yeah, they are. Yeah. So that's a good point, Dave. I think Marcus's studies, his were 4 months in duration.
Okay, I mean, that's decent.
Yeah, that's pretty decent. Our two studies, so Cody's study was 6 weeks. Now, keep in mind, he recruited previously resistance trained individuals for his study. And then our other study was done by Brooks Mobley, who's now at Kentucky, but we did a 12-week training study there. So that's a fair point. Cody's study in particular, 6 weeks, one can make the argument that, well, perhaps the low responders may have started responding after that time point.
Right.
We think 3 to 4 months is a good amount of time. And I tell people, look, because we get a little bit of pushback on this, but like everything else, I mean, it's a bell-shaped curve in terms of the respondents, right? You're going to have people that are there towards the far right, which are your extreme responders. You're going to have a lot of people in the middle, which are going to be kind of your mediocre responders. And then there are always going to be people that are there towards the left end of the curve, which are your low responders. I mean, that's pretty much how everything works. So to us, it's not shocking that we see that. We're just trying to understand the molecular reasoning behind that, right?
Sure.
Yeah.
Yeah, I mean, I know recently there are a few higher volume studies that have been put out, and some of them have concluded that the higher the volume, the fewer number of non-respondents there were. So just thinking that maybe like in endurance training, they just need more of a stimulus. So for one, what do you think about that? And two, how was the volume in these studies you're referring to where you had non-responders?
Yeah, so the volume in the first study, which was our untrained individuals, and they had trained for 3 months, they worked out 3 days per week, full body. 1 day was a hypertrophy day. So for every exercise, everything was a compound lift, right? So bench press, bent over row, back squat, deadlift. They were doing 4 to 5 sets of 10 repetitions on Mondays. Then they would do Wednesdays, which would be strength, right? So strength was 5 sets of 3 to 4. And then Fridays, they would do 6s, which is kind of a hybrid day. So I wouldn't call that extraordinarily high volume. I mean, that's typical of what you would see with undulating periodization. The second study, which I don't know if Cody talked about his dissertation, that was his dissertation, it was extraordinarily high volume. So, you know, they were doing kind of the same workout scheme, 3 days per week, all compound lifts. During the 6th week, let's say you were just looking at the last day of training for the entire study, like, you know, you finish that day and you're done with training, right? All the subjects were doing 12 sets of 10 reps on back squat, 12 sets of 10 reps on bench press, 12 sets of 10 reps on stiff leg deadlifts, and then 12 sets of 10 reps on supinated or reverse lat pull downs. So that's everybody in that study, and yet we did have some non-responders.
That was per week, or that was each session?
That was the last training session. That last week looked a lot like that. So they would do, we call them 12s because it was 12 sets of everything, 10 reps. So that last week during week 6, it was 12s on Monday, 10s on, 12s on Monday, 8s on Wednesday, 12s on Friday.
Wow.
Now, every week it was a progression in volume. At the beginning of the study, it was 4s, 2s, and 4s. Right. So that means that for every lift, on Monday, they were doing 4 sets of 10. Then on Wednesday, they were doing 2 sets of 10. And Friday, 4 sets of 10. And then from there, we progressed volume, right, to the point where it was 12s, 8, 12s.
And this was over 6 weeks.
This is over 6 weeks. And these people were previously trained.
Yeah, I mean, it's a short duration, but I mean, that's still pretty damn high volume, especially for those types of lifts.
Very, very high volume, for sure.
And still non-responders.
And there were still non-responders. So I will say this, when I was talking about this gets a little bit complicated, right? Because we have 2 studies. Both studies were different in duration. And then the first study was in people that were previously untrained or they were not well trained, whereas the second study, these people were trained, right? So we're talking about two completely different studies practically. In the first study, ribosome biogenesis was important. So in an untrained person, your ability to synthesize ribosomes with resistance training is going to, appreciably affect your anabolic response, is what we see. In the second study, in the high volume training, previously untrained, et cetera, ribosome biogenesis did not play a role because both the low and the high responders saw an increase in ribosome biogenesis. So,
to the same degree.
To the same degree, right. So we looked at a slew of other markers. We looked at markers involved with proteolysis, or the breakdown of proteins, because we thought, well, man, low responders, they just may not be recovering from this high volume workout. Didn't see any difference between high and low responders with those proteolysis markers. Same with muscle damage markers. So serum CK activity wasn't any different. The one thing in study 2, which was the 6-week high volume study in trained individuals. So the one thing that we found, which was predictive of the training response, was the percentage of type 2 fibers. So if you had more type 2 fibers, you were typically a higher responder. And then if those type 2 fibers were smaller at the beginning of training, you typically had a greater response.
Right.
So if you are a person that has a lot of type 2 fibers that aren't very big to begin with, then you would respond to high volume training if you were previously trained. That's me trying to translate the finding, right?
Yeah. So, and I brought this up to Cody too, and it's interesting because in one sense, it makes sense. Like if everybody had a similar genetic potential, then those who had these smaller fibers would have more room for growth, right?
Sure,
and then so then they could grow more.
Yep.
However, I would think that, because I think the average training duration was about 5 years in this study, right?
That's right. Yep.
So, if you broke that up relatively evenly, I would think that the people who were bigger, assuming their training backgrounds were similar,
Yep.
I would think that people who were bigger had better genetics. If they're doing something similar, they're already bigger, probably better genetics. So I would then think they also have more room to grow just because of their increased potential. Does that make sense?
Right. Yeah. So what our take is that maybe they have almost reached a natural ceiling, if that makes sense. So we're kind of thinking about it in a different way, right? And that's assuming none of these guys are taking steroids or chemically enhanced.
Right.
So the bigger guys, we think, are closer to that human potential, or their individual, their individual limit, if you will.
Right.
Whereas people that are not as big still have a little bit more room to grow. And we thought that high volume training kind of forced the issue, if that makes sense.
Right. And I guess the argument there would maybe be that, assuming they had similar ceilings, I would agree. Obviously, the ones who are smaller have more room.
Yep.
I guess maybe the variation between those two groups then is that, I've seen people argue that two people might reach the same size, but one can get there in 5 years, and for one it takes 10 years. So maybe in that bigger group that didn't have as much of a response, they had a bigger initial response in their first few years of training, whereas the other people were maybe more slower growers.
Sure. Yeah, that could be it. We didn't track them for 5 years.
Yeah, right. Yeah, who knows? So other than that, what other variations do we see in genetics in terms of what could affect muscle growth? Anything else that we see that's really significant?
Yeah, so those are the two things that we saw. What I just told you was, in untrained people, the ability to synthesize ribosomes seems to be important. And not only have we shown that, but Marcus Baumann has showed it. Andre Figueredo, who was in David Cameron Smith's lab, also showed the same thing. So we think ribosome biogenesis, as you start training, is important. Beyond that, there really hasn't been much literature in this area. So I wrote a review article, and all of my students, including Cody, kind of helped me synthesize the literature. Marcus Baumann has directly looked at this question through a few studies. So what's interesting is that in our studies, as well as Marcus's, Self-reported food log data suggests that there are no differences between high and low responders, and that includes protein intake, total kcal intake. Yeah, exactly. Now, I always tell people that doesn't suggest that if you give low responders more calories or more protein, that wouldn't enhance the response. But what we're saying is that the response wasn't due to self-reported food intakes, right?
Now, during these studies, are you guys telling them just eat as you have been eating?
Yeah, so Cody did a really good job of making recommendations, and his recommendations, the intent was to make sure that they were in a caloric surplus because of the high volume.
Right. Okay.
Additionally, they were getting supplemental protein to kind of support the growth. So all of that aside, when you look at high and low responders, yet again, we didn't see any differences in protein intake as well as calorie intake.
Did they gain similar amounts of weight, and the non-responders just gained almost entirely fat?
No. So, and I got to pull up the data to look at that. So we did whole body lean mass composition and fat mass composition using DEXAs.
Okay.
So we broke that out into fat mass as well as lean mass. Now, I don't know that data offhand, but there wasn't anything striking with regard to fat mass changes between high and low responders. So that's not saying that the low responders are gaining weight and it's all in fat. We're not seeing that, if that makes sense.
Okay. Well, I'm just wondering, so they must have not been in much of a surplus if they didn't have lean mass gain or fat mass gain.
Yeah, and therein lies the problem, is that Cody made the recommendations, right? But they all, we think that looking at their numbers, I mean, they weren't consuming 4,000 calories. They were 2,500, 2,600. So even though Cody did a really good job making recommendations, they were only eating so much.
And that's what I wonder, because, I mean, nobody gains 100% fat, right? I mean, even fat tissue has been found now to have some lean component to it. So if you have people gaining like 20 pounds over the course of a year, I can't imagine they're not putting on any muscle mass, but obviously there's going to be huge variations in the degree to which they do put on that mass, the muscle mass.
Sure.
Have you looked into anything with the androgen receptor density?
Yeah. So I was getting back to, other genetic components which may delineate responders versus non-responders. So besides Marcus's data and our data, Stu Phillips has done a lot in this area. Rob Morton's his PhD student, and he recently put out a paper saying that if you have a high level of androgen receptor in the muscle, you will be a high responder. And I believe that study was in previously trained males as well. So it was like Cody's study. So we actually tried to replicate that assay. We did Western blotting, and we validated our antibody through the appropriate means, for lack of a better term. We didn't see anything similar to what Rob and Stu reported. That's not to say that they're wrong. It's just, at least in our cohort, the androgen receptor doesn't play a role. And that is for both studies, right?
Wow.
So the relative amount of androgen receptor, whether you're untrained or trained, doesn't drive the anabolic response, according to our data. Rob's paper seems to suggest that it does. So like all science, right, you're going to have divergent findings. I think it's something that definitely could be contributing. We just need to try to figure out, if it's a thing or not with more research.
Yeah, that's interesting. I had thought most of the studies on it had shown that androgen receptor density was a pretty important factor. I didn't actually realize that these two recent ones show that it was not a factor.
Yeah, so the only Stu has, I think, 2 papers now showing. One was showing that the change in androgen receptor positively associated with the change in muscle hypertrophy. And that was by Cam Mitchell, I think, and +1. And then Rob's most recent data, they actually compiled a bunch of participants from multiple studies, and they showed that both at the pre-intervention time point as well as the post-intervention time point, that the high responders had more muscle androgen receptor.
Right.
Our data doesn't say anything to that tune, right? So,
Yeah.
in study number 1, which is 12 weeks untrained people, in both the high and the low responders, we saw a decrease in the androgen receptor protein with training, okay? And we said, well, to us, this kind of makes sense, because if resistance training is operating in part through testosterone signaling in the muscle to facilitate adaptation, then there could be this compensatory downregulation in the androgen receptor, right? You increase the signaling, you're going to potentially downregulate the receptor.
Right. Interesting.
In study number 2, which is Cody's study, we didn't see any change. And so then from there, we bought a positive control, which was a cancer cell lysate. It was a prostate cancer cell lysate. We did a Western blot on that to make sure that our antibody was valid, right? Because that's critically important with Western blotting. And it indeed was valid. So we also supplied that with Cody's data saying, look, we understand that some people are saying this, that androgen receptor protein levels are important. We're not seeing it. Now, that's not to say that signaling, there's a difference between looking at the protein content of a receptor in muscle versus looking or interrogating how that protein functionally operates, right? So I think to test that question, you would have to design a study that is long-term to say these are responders, these are non-responders. You get like a pre and a post-intervention biopsy. But then there at the very beginning, you perhaps do a bout of training and you get a kind of a series of biopsies following that first bout of training, because from there, you can look at the androgen receptor in terms of where it's localized, right? Does it move to the nucleus to enhance its effects on the genome, right? Because that's how androgen signaling operates. It binds as a transcription factor to turn on genes related to cell growth. So I think you need to do that to really start parsing out the importance of the androgen receptor in terms of the hypertrophic response.
Sure. So I find a lot of this stuff very interesting. I think a lot of listeners do find it interesting. We also like practical takeaways from this. And unfortunately, as of now, there's not a ton you can do to change your genetics. But maybe we hope that there's things that we can do in our training or diet or habits that, if we don't have the best lot genetically for this, are there things we can do to tip it in our favor? And of course, we know obviously like the basics of lifting and nutrition, but is there anything that you found that the low responders might be able to do to again kind of improve their lot?
Yeah, so right now it's all been exploratory on our end in terms of just looking for biomarkers that delineate high versus low responders. Moving forward, what an idea we've been kicking around is, replicating these studies, identifying the cohorts with ultrasound in terms of who grew muscle and who didn't, and then from there, altering the training, right, of the low responders so that perhaps you add more volume. Perhaps you do some sort of concurrent training, because there is literature suggesting that high responders may have more muscle capillaries, so the number of capillaries surrounding muscle fibers, and that in and of itself is very interesting. That comes from Tim Snyder's and Luke Van Loan. But that to us suggests that quote unquote nutrient delivery to muscle may be driving the anabolic response, right? If you have this increased capillarization. So the short answer of it is right now it's kind of it's not ready for prime time in terms of making recommendations, right? Right now we're just trying to figure out molecular markers that delineate the clusters. I think moving forward, one of the goals of the lab, and this is going to be like a 5-year goal because these things will take, they take a long time. But moving forward is, can we have somebody walk in and say, boy, you're not going to be a good responder, right? Whether we put an ultrasound on the leg and say, according to your muscle thickness, and all of our past data, it just doesn't look like you're going to be a good responder. And then from there, can we put you on a some sort of exercise prescription which optimizes that training response. We're not there yet by any means, but I think that's where the field obviously needs to head, and that's kind of part of the reasons why we're doing this stuff and starting the research, right? Kind of in the first phase of that overarching goal.
Right. So that's, like you said, kind of like a 5-year goal. What are you thinking? Like, what's the research you're actually looking at right now, and like, what are you hoping to look at maybe in the next year or so?
Yeah. So the research right now is actually, I think it's peripherally involved with the high versus the low responder. But the research we're looking at now is another thing that we found with Cody's data in his dissertation, and it deals with sarcoplasmic hypertrophy. Have you guys talked about this at all on the podcast?
I think he and I might have started talking about it. Yeah, we did actually. Yeah, we did. Yeah, but not. Like, feel free to go into it.
So what's interesting is that the high responders in Cody's study, we continued to analyze their muscle tissue. And we found that while the muscle fibers grew, we saw a decrease in the amount of actin and myosin content or concentration in that muscle. So we followed that up with a histological interrogation, which confirmed our earlier finding that yes, actin and myosin are going down in terms of protein concentration. And so to us, that's saying as the cells were growing, there was not a proportional increase in the amount of contractile protein. What we're not saying is, these people are not losing contractile protein, but what we think is happening is that the cell growth is outpacing contractile protein accretion. And so we're calling this sarcoplasmic hypertrophy, right? Because the sarcoplasm is the fluid around the contractile elements inside of a muscle cell. Mm-hmm. So we're trying to sort that out now with Chris Van. He's in the lab. He's a doc student. Now we're designing a study to see if this is a thing, because with Cody's data, it was more or less an unanticipated finding that left us scratching our heads saying, wow, is this really happening? Like sarcoplasmic hypertrophy with really high volume training? So Chris Van, what he's going to be working on is unilateral leg training. He's going to have individuals train one leg with very high load training. Mm-hmm. And we're going to be taking a series of biopsies, doing MRI scans, et cetera. With the other leg, they're going to be doing very high volume training. And what that will allow us to do is see if muscle cells grow under both circumstances, is the high volume growth due to sarcoplasmic hypertrophy, whereas the high load growth, is that due to myofibrillar hypertrophy, or the increase in cell size with a proportional increase in contractile proteins. So that's one of the things that we're working on right now. We are actually working with Stu Phillips. He's sending us some tracers. He's on Chris's committee, so we've had really good interaction. Stu's skeptical. There's other people that are skeptical, and I just say, look, we're just trying to figure out if this is a thing, right? I mean, I think there are examples of it. You can clearly make a case for creatine, right, in terms of the rapid lean body mass gains that you see in the literature. In our head, at least, that's not all contractile protein, right? There's going to be some fluid shift involved. Anabolic steroids, I think, if you talk about D-ball or things that rapidly put on mass, I mean, one could make the argument that that's not all functional contractile protein in terms of the lean mass that's gained. I mean, some of that is the muscle cell expanding through sarcoplasm. And so, for us, it's not a far stretch. Look, if the muscle can undergo this process with these other things, then pumping up muscle with high volume training may operate through a similar fashion.
Right.
So that's something that we're gonna test out here this year. We'll find out.
Okay. So your hypothesis would be that the higher volume group would have higher growth due to sarcoplasmic hypertrophy.
That is correct. Yeah.
Cool.
Yeah.
Cool.
Now, so that's an easy hypothesis because there's impetus in the literature as well as our own data to support that. What we're unsure of is whether or not high load training, if it does cause hypertrophy, is that due to this expansion of the contractile apparatus? Because mechanistically, we really can't explain why that is. Then we're going to have to try to figure that out, right? I mean, it all gets down to how different loads, I think, talk to your myonuclei, right? The stressors involved, the metabolites accumulated with high volume training. You know, do these things, whether you do high load versus high volume, are there different molecular pathways in muscle? Are there different genes that are turned on in muscle in response to both of these, which ultimately drive the two different phenotypes, right? So yet again, this is going to be a multi-year sort of interrogation.
Right. Yeah, that would be interesting because, I mean, back when I was like starting training 15 years ago, and you would see that on some of these fitness articles where they'd say, you know, the heavy training is what really like builds up the muscle tissue. And then there's the sarcoplasmic hypertrophy from these higher rep training sessions. And people would, I don't know if you know Dr. Scott Stevenson.
Mm-hmm.
So he was somebody I've known for a few years now. And I remember him saying, well, where are you getting that from? You know, we don't actually have evidence of sarcoplasmic hypertrophy, but everybody's always talking about it.
Right.
And now I guess we finally are maybe getting into that realm.
Yeah, no, I agree. I think the way we're looking at it is other people have shown it. It's been kind of sporadic in the literature and no one's really put a name behind it. And so we were kind of the first to put our flag in the sand and say, yeah, that's what we're observing. We're observing sarcoplasmic hypertrophy. Now, the trick of it is the methods used to assess it are delicate, and you have to use multiple methods. So we're using a very easy method relative to other things that can be done. We're basically looking, if you take the muscle tissue out of the leg, you homogenize it in a tube. You have to use special buffers to get the contractile proteins versus the sarcoplasmic proteins. From there, when you get the contractile proteins, you simply run it on a SDS page gel, which is, you know, common in any wet lab, and then you stain that gel and you get the density of the actin and myosin bands. You then normalize that to the milligram of tissue you put in. So if that value goes down, that tells you that you have a dilution of the actin and myosin based on the amount of tissue that you put in.
Yeah.
And so that's exactly what we observe. Now, from there, we're going to try to get a little bit more sophisticated because we're going to use phalloidin staining, which looks at muscle cells in cross-section, and it tells you the amount of actin per muscle fiber. And then we're also going to try to use electron microscopy, which really gets down into the details, and you can look well within individual muscle fibers in terms of the sarcoplasmic space as well as the contractile protein space.
Yeah, I mean, that'll be cool to see. One other thing that Cody and I had talked about was maybe the reason behind that, because I know something I brought up to him was years ago, there was a guy I knew, but some people talked about where they would say, you know, after you've bulked up, don't just cut right away, but hold on to that size. And they would talk about like solidifying your gains. You know, and there wasn't really a lot of science behind that, but Cody and I were talking about maybe that is a mechanism by which that makes sense, is if the sarcoplasmic hypertrophy is almost like making room for the other like actual protein units. I don't know if that makes sense to you or not, but that's something that we had kind of talked about.
Yeah, and that's what we put in the paper. So the paper, I'll shameless plug for this paper. It's on +1. Just search sarcoplasmic hypertrophy. So we think, and a lot of this was Cody's idea with the bioenergetics of it all, but we think that if you do high volume training, what you're doing through sarcoplasmic hypertrophy is spatially priming muscle cells to then lay down contractile proteins eventually, if that, you know, with more longer term training.
Right.
We also see an upregulation in glycolytic enzymes and other enzymes used to synthesize ATP. Well, given that myofibrillar proteins are very large and they require a lot of ATP to synthesize, that also makes sense in terms of this priming mechanism that you're talking about. So yeah, I definitely buy into that idea for sure.
Cool. So if people want to find more of your work and your studies and keep up with you, should they just be searching PubMed? Is there anywhere, like you have a web page or anything like that people can check out?
So I have ResearchGate I don't. So you just Google ResearchGate, and then you type in Michael Roberts Auburn or Mike Roberts Auburn. Y'all can see all the most recent articles on that. Trying to think if there's any other. I think that's probably the best website to find all of our research articles for sure.
Okay, awesome. And I'll put a direct link to that in the description of the podcast, and so people will be able to find you.
Sweet.
All right, man. Well, thanks again for talking today.
Dave, thanks for having me, man. Appreciate it.